So Dr. Miner is professor of medicine and nephrology at Washington University School of Medicine in St. Louis. He's using several mouse models of Alport syndrome to search for pathways that injure the kidney, to test the effects of drugs that inhibit injurious pathways, and investigate innovative therapeutic approaches with funding from a new 5-year NIH grant entitled Innovative Approaches to Treating Alport Syndrome. He's president of the American Society for Matrix Biology and a very dedicated Alport researcher. Thank you.
Thank you, Lisa, for that introduction. Thanks for having me here also. I'm a professor, I'm a scientist, I'm a teacher, and there's almost nothing I enjoy better than educating patients like yourselves as to why your kidneys aren't doing so well. And I hope I'll get that across in the short 12 minutes that I have. So, just a little bit about the kidney and the nephron. Kidneys look like kidney beans, they really do. I don't know which came first, the kidney bean or the kidney. Most people have two, some only have one. The kidney has, part of it does the filtering and the making of the urine, and that urine then flows into these little compartments that then lead to the ureter that pumps all the urine into the bladder, where it's held until it can be eliminated appropriately. This is one of the millions of nephrons that humans have in their kidneys. Each kidney has about 1 million nephrons. It's a very complex little structure. Each of these millions of nephrons is its own little microfiltration unit that makes a tiny bit of urine. That's all added together in these renal calyces that then ends up in the bladder. So it's important that all of those nephrons are functioning. The glomerulus, this part that's affected in Alport syndrome, is what does the filtering. The tubule, the rest of the nephron, turns that primary filtrate into the final urine that makes it into the bladder. And I'd be happy to answer questions later this afternoon, as Lisa indicated. So this is now a schematic diagram of this glomerulus that does the filtering. The blood is flowing through these cross sections of capillaries. The two components I want to mention the importance of are the podocyte, which makes the glomerular basement membrane. It's the glomerular basement membrane that is not normal in Alport syndrome. The podocyte becomes dysfunctional when the basement membrane is abnormal. As podocytes are somehow injured by that, and they will eventually fall off and enter the urine, show up in the final urine, and it's the loss of podocytes that leads to loss of glomerular filtration and your reduced EGFR. I'm sure many of you know what that is. So I have a movie now made by some investigators at Indiana that demonstrates how the glomerulus filters. And the glomerulus exhibits glomerular perm selectivity. And so what these investigators did is they took a rat and they put it on a microscope stage and exposed the kidney of the rat outside the rat's body and put a microscope objective on top of that kidney and injected fluorescent tracers into the bloodstream of the rat. And so what you're going to see in this movie that I hope will play, I have to exit unfortunately, is that so used to a Macintosh. Here we go. Okay, the yellow molecules are large, the red molecules are small. And so you're seeing the yellow molecules remain in these functioning capillaries, whereas the red small molecule enters the urinary space and flows down the tubular portion of the nephron, where it would normally become part of the urine of the rat. So this exquisite selectivity of the glomerular filtration barrier is what's defective in any patient with glomerular disease, and also in patients with Alport syndrome are included. Yes, got it. So moving on, I'm going to give you a bit of an analogy, which I like to use when I talk to patients, as to how that glomerular filtration barrier works, because it's so critical. Think of it as similar to a coffee filter. I even use this analogy to scientists in some cases. It acts just like a coffee filter. Coffee filters at high magnification, you can see little pores in the coffee filter. What do those pores do? They allow the passage of water and small molecules such as caffeine, flavor, color, notes of toffee and walnuts in some cases. But those pores also restrict the passage of the grinds into the filtrate, which is the coffee. The filtrate is the coffee that we drink, right over here. Excuse me. In the kidney, the glomerular filtration barrier allows the passage of water, small molecules, salts, proteins, small proteins, and nitrogenous wastes that become the final urine. But that filtration barrier should restrict the passage of blood cells and proteins, large proteins, into the filtrate. So you can see in this electron micrograph, a low power micrograph of the glomerulus, the proteins are mainly at high concentration inside the bloodstream, but at low concentration outside in the urinary space. So here's now a higher magnification view of that filtration barrier. We have the blood flowing through the glomerular capillaries here, the filtration barrier, which has 3 layers. I've only mentioned the glomerular basement membrane, identified by the red arrows, and the foot processes of the podocytes, which then, this is the urine, will then go on into the tubular portion of the nephron. So in Alport syndrome, the most obvious indicator that a patient has it is this glomerular basement membrane becomes split and thickened and has kind of a moth-eaten appearance in this particular case. Compared to the normal basement membrane should be very smooth and ribbon-like, whereas in Alport syndrome it becomes widened and thickened and split, and that's typical, very typical of Alport syndrome. That's how some of you were probably diagnosed by your pathologists. So I want to briefly mention a bit of a complex topic is Alport syndrome and the genes and proteins that are involved. I know many of you know that you have a mutation in the COL4A3, A4, or A5 genes. There are actually 6 collagen 4 genes, alpha 1 through alpha 6. But the reason mutations in alpha 3, 4, and 5 cause Alport syndrome is that that's the main component of the GBM, the glomerular basement membrane, which is the main part of the filtration barrier. So let's focus on the alpha 3, 4, 5 genes. The alpha 5 gene is on the X chromosome. Males have only 1 X, females have 2. So males that have an alpha 5 mutation don't do as well as their female relatives who might have the same mutation, because the females will have another X chromosome that can take over some of the job. So that's why mutations cause the most prevalent form of disease, and boys are more severely affected than girls. But for the alpha 3 and alpha 4 genes, those are not on the X chromosome. They're called autosomal genes. And homozygous mutations, meaning both copies, so we all have 2 copies of alpha 3, 2 copies of alpha 4. Both copies of one of those genes needs to be affected to cause the severe form of Alport syndrome. So boys and girls are equally affected because there's no sex linkage. So there are great animal models for Alport syndrome researchers like myself. There have been several naturally occurring dog models of this disease that have appeared in the literature. Veterinarians are very good doctors for their patients, and so they will follow up to find out why their patients have kidney disease. And so there's a colony of dogs at Texas A&M, carried by Mary Nabedy, who's an Alport syndrome researcher. There are multiple mouse models that I've made and other laboratories have made. There is a zebrafish model of Alport syndrome that can be used for studies that I won't get into, but zebrafish also have kidneys. They're widely used in genetic research and developmental biology and can be used for Alport syndrome research. And then there's the fruit fly. The fruit fly is one of the oldest models for genetic research that researchers have used. Fruit flies have a cell that's very similar to a podocyte. It makes a basement membrane that is somewhat similar to the glomerular basement membrane, but the important thing is it has collagen 4, and so one can use fruit flies to manipulate their collagen 4 genes to test the impact of mutations that some of you in the audience might have to determine how pathogenic those mutations are. I can talk later about this if you're interested. So these are useful for understanding disease progression, testing drugs for safety and impact on disease symptoms before testing drugs in humans, exploring the impact of genetic variation and environment on kidney disease progression. They're useful for investigating the pathogenicity of variants of uncertain significance that are found in patients suspected of having Alport syndrome, but it's not clear that that's really causing their disease. And also for investigating emerging novel therapies, such as protein chaperones that some researchers are using to try to help defective collagen 4 proteins fold properly, for replacing proteins that are defective in Alport syndrome. They're useful for gene editing approaches. CRISPR, some of you might have heard of, won the Nobel Prize 3 years ago for those who discovered it. RNA modifications can be done to try to promote or try to bypass nonsense mutations, as well as nonsense codon read-through approaches that my lab has been doing using our mouse models. So one thing about Alport syndrome and other glomerular diseases is that we know the defect is in that collagen 4 component of the glomerular basement membrane, yet the disease spreads to the tubular portion of the nephron, and the entire kidney becomes fibrotic. It's not just restricted to the glomerulus. So why is that? Why does it spread to the tubular interstitium compartment? It's important to figure that out, because if we know why, we might be able to find a drug that can stop the process of the disease progressing from the glomerulus into the rest of the nephron, because that would slow nephron deterioration and keep that EGFR number higher for longer. So we had a hypothesis years ago that albumin in the urine, the proteinuria that some of you have and know about, is not just a sign of kidney disease, it also can promote kidney disease progression. And so we tested this hypothesis by making mice that don't have any albumin. They seem perfectly normal. We bred those mice to mice that have Alport syndrome, so that we have mice that have Alport syndrome but no albumin in their body. These mice live a lot longer. So the black curve is showing you those mice compared to their brothers and sisters and cousins that do have albumin and Alport syndrome that reach kidney failure between about 250 days of age. We take away albumin, some live to over 400 days of age. So is this going to help anyone in the room? Hopefully. We have a hypothesis that fatty acids that albumin carries normally in our bloodstreams can injure the tubular portion of the nephron. And in Alport syndrome, too much of that albumin is crossing the filtration barrier, getting into the urine and injuring those cells. So we're working on ways to try to figure out how we can understand why that happens and then slow that process. So thanks for your attention. These are some members of my lab who did some of the work I talked about. Thanks so much to the funders, NIH, the Alport Syndrome Foundation especially. We're all wearing shirts with octopuses because the octopus is the mascot of the podocyte community, because podocytes have multiple arms that sit on the glomerular capillaries. And congratulations to you all for being here. Those of you who have never met an Alport patient before, you're going to learn so much from them about how to deal with this disease. And the rest of you, you need to help those people through your own experiences. So thanks for listening. I look forward to questions later today.
Thank you. Now we've got a great overview to get started, right? And now we're going to talk a little bit about the future and some really cool stuff that's going on at Dr. Perin's lab. Dr. Laura Perin is an associate professor at the Keck School of Medicine at University of Southern California. Her research focuses primarily on studying mechanisms that regulate kidney damage, with specific attention to glomerular injuries and renal cancer. Dr. Perin completed her PhD in stem cells and tissue engineering at the Harvard School of Medicine and obtained master's and bachelor's degrees in biological sciences at the University of Padova in Padua, Italy. I also want to note, by the t-shirt too, you can tell, Laura Perin, Dr. Laura Perin, is also a living donor hero. She saved her husband's life through living donor kidney donation. and has become such a great advocate for it that she wrote a book. She shared her experiences and also interviewed a lot of other patients who have been kidney donors and asked them about their experience, how it went, why they did it, what their life is like afterwards. I find it pretty extraordinary. And so she has some free copies for us, for those of you who are interested. They'll probably go quick, but back at our ASF table back there, and you can also get it on Amazon. But it's a beautiful book. I gave it to my living kidney donor son for his birthday. All right, here's Laura Perin.
Good morning to everyone. It's a great honor for me to be here today. And I'm a scientist, but I'm also the donor, and I understand living with someone that has kidney disease and how hard it is to live with the loved ones that have this disease, and how it impacts everyone, not just only the person who has the disease. So today I'm talking to you a little bit about the research that we all do in the laboratory. So this is how you kind of see the kidney. You have your beautiful kidney, and then when you have Alport and you're sick, you just see it with the thermometers. But this is how scientists see the kidney, and Dr. Miner gave an incredible explanation about the kidney works. And so when I think about Alport, we just go like to the glomerular, what's happening to the podocytes, and what we can do to save a podocyte life. Those podocytes is our patients under the microscope practically. So we have a lot of tools in the laboratories, and our main goal is to really protect podocyte and protect the glomerular basal membrane, and also another cell type that is on the other side of the glomerular basal membrane, which is the endothelial cells, so the capillaries that are within the glomeruli. So our work, what we really wanted to do is to really stop to have a fibrosis and to have a scar tissue within the glomeruli. And so we wanted to establish back to the normal and avoid the disease progression specifically in Alport. So these tools are extremely important for us because they help us to understand how Alport syndrome develops and how Alport syndrome progresses. And also it's extremely important because if you understand the mechanism that regulates this progression, we can use this knowledge to develop a new therapy that potentially one day can help one patient to really to find a cure for this disease. And you can see in the circle on the left most of the tools that we use. And so I will try to really explain in a very simple way, hopefully, what we do in the laboratory and which are these tools. So one of the most important tools that we have in the laboratory are the cells. Cells are important because we can study how the podocyte, how the endothelial cells work, and why they get damaged. We understand how they respond to toxins and drugs. We can build more complex structures starting from one cells and let them grow and form a small kidney that I will show you afterwards. We can study how we can modify their code to make them to feel better. We can also change their environment to see if we can avoid it to get to further damage. So this is just an example of the cells that we use. For example, in our laboratory, and I have Stefan and Sarkis here that work with me in the research, in the Alport research. We use cells from the kidneys that, for example, they're not used for transplantation. So we bring them in the lab and we isolate them and we use them under the microscope to do all our experiments. Some of the cells are also present in our urine. They can, be shed in the urine. So we collect them and we use in the laboratory. I really want to thank here personally all the Alport mom that are helping my research in donating their amniotic fluid because that is a very important source of cells that we use in the lab. And also the induced pluripotent stem cells where we can modify these cells and program and differentiate it in what we want. So these are very important tools for us because these cells then are applied in a different other approaches. And I'm going to show you singly all of them. So one of the things that we use in the laboratory are these organoids. So we are trying to build mini kidneys So we can use them and study how Alport developed. Think about the mini kidneys as a, you have your brick to build a house, and you keep, the cells keep replicating one on top of the other, and in the end, you really form these mini kidneys. Dr. Lennon, which is an incredible Alport scientist and researcher, has really developed this Alport mini kidney, and she uses it, for example, to understand how the collagen 4 develops. Another group has also developed and just published that they're generating a mini kidney with the Alport 5 collagen mutations. And they're very important because we really can understand step by step how the kidney is developing. So we can have a lot of knowledge about that. The second one are the extracellular vesicle. This is specifically something that Sarkis and I are working on. Think about extracellular vesicle like little messages that you can find in a bottle. So like real messages, the extracellular vesicle can tell to other cells what to do, ask for help, or inform about something. So all the cells in our body produce this little vesicle, which are called, we call it extracellular vesicle, microvesicle or exosome, and they contain a lot of information inside, like the messages on a bottle. And so you can isolate this vesicle, and you can see if they have a therapeutic approach, for example, in establishing podocyte biology or structures. This is, for example, was one of the work that Sarkis and I were funded by the Alport Foundation, and we have proved that in animal model, if we injected this vesicle, we can prolong a little bit the lifespan of these mice and slow down the progression of the disease. We also know how they seems to work, and if you have any questions, I would be happy to answer to all of them later. Another tool that we use in the laboratory are this fancy microfluidic system, this word that seems to have a lot of impact now in research. And this is also, so try to imagine, for example, to paint a little kidney within a white rice, how small it is. So what we are doing in the lab is we're really recreating this small glomerulus between two glasses. And so we can do a lot of experiment with that. So you can study, for example, if you have a collagen 4, if it's mutated, what is happening to the cells. So this are done with human cells. So in conjunction with mouse study, we can also look at what's happening to the human cells. This is, for example, the chip that we use in the lab, and this is the work that Stefano has done, and then he has also an NIH-funded grant about that. It's like these little tiny glasses, and we have, you can see here in the picture, color is the mouse. Well, you can see in blue and red, we have the podocytes and the endothelial cells, and we form the Alport glomerulus, so we can test a lot of different things in our studies. Then another important tools that we have in the laboratory is the gene editing. Think about, your kids when they're building with the blocks, they're building their house, they don't like where the door is or the window is, you just move it around, and you put something, you move a space, and also you move the color of it, and so you create the house as you want to. So this is what scientists are trying to do too with their, you can modify a gene expression or change and do a lot of different things with this tool that we have in the laboratory. And so in the Alport setting, we wanted to correct the bronchocollagen. Dr. Meine is working on a project funded from the FASN, and there is an aim to investigate when during kidney disease, the patients can benefit from a change with the CRISPR technology to fix the mutated collagen. And there are also other groups around the world in different countries that are using this gene editing technology to modify and restore the collagen 4 expression. So then there is another fancy word that we use, which is called spatial transcriptomics or multi-omics. And in the last 5 or 10 years, I think it's really, it's getting into in all the laboratory more or less. And this omics analysis is very important because it allows you to collect information about genes changes and protein change in the context of Alport, which I'm studying in the lab, so we can really understand the step of the disease progression. And that is really important, again, because it will help us to design specific therapeutics that can help our populations. I'm not going into details about all of this technology. Sometimes they're very complicated for me to understand, but I give you some example. For example, using one of these technology, Sarkis has found that not all the endothelium cells or the capillary cells that form the capillary and the glomeruli are the same. Based on their positions and based on the disease progression, these cells changes. So if we can really understand all of these paths, maybe we can really find when we need to intervene, what we can modify to make them to feel better, to go back to their natural state. Another project that I'm really involved to is what is called the spatial transcriptomics. Think about that this morning driving here, you got your map to go to the Alport Connect, and instead of even street maps, we have gene maps. So we take a human biopsy from Alport patients, and then with this technology, we can really find each single where the things are expressed, and we can design our map of the glomeruli. And doing this, we have, for example, found that some Alport patients has immunocells within the glomeruli, and we were wondering why. This is just the collagen disease, why we have also these cells that are going around in the glomeruli. But this technology really is helping us to point out all different cell lines. Dr. Lennon, instead of working with the genes, she's working with the protein, and she's working on the matrizoma project, which is an extremely very important project because it's also analyzing with the omic approach not only the collagen 4, but all the other proteins that are a part of the glomerular basal membrane. It's not only the collagen, and so she's looking into the human Alport patients, and she really has found a lot of different other proteins that might contribute to the disease progression too. And the last one is also, we use a lot of microscope, and there are very fancy and very expensive microscope that allows us really to look not only the big pictures, but really going to in details about how the podocyte is made, and not only outside, but inside, and looking all the, it's like think about having the radiology of the podocytes in the same way you have an MRI when you go to the doctor. And this is just a beautiful pictures that Dr. Miner shared with us, where you can really see, you can see there in green the collagen 4, and in red another type of collagen, and it's made with this picture is taken with a very technique which is a super resolution imaging. I don't know how to use, Dr. Mane knows how to use it, but you can see very well, I have no idea, but it's actually really cool, or it looks very cool. So it could be a painting that you can put in your walls, because I think the colors are amazing. So between, the animal models, the microscopy, the cell lines, all of that, we are using, most of them in our laboratories, and this is with the final aim to really find ultimately a cure for Alport syndrome. And later I would be happy to take any questions, and I'm here all the day. Thank you.
A lot of expertise in this room, including our next speaker, who's going to get more into the details of why we're collecting human data from all of the folks like you, like us. Dr. André Weinstock has a BS in biochemistry and a PhD in analytical chemistry. He serves as a volunteer research director for Alport Syndrome Foundation and serves on our board. His mother, sister, and he were all diagnosed with Alport syndrome in the 1980s. André spent 2 years on hemodialysis prior to receiving a kidney transplant from an anonymous, non-living donor. He's tried to make the best of this gift and spent the last 2 decades traveling the world, finding love, and building a career as a research scientist in the field of pharmaceuticals, medical devices, and diagnostics. And André is like extended staff for ASF. There isn't a day of the year that we're not on the phone or by email. He's an extraordinary volunteer. So next, Dr. André Weinstock.
Thank you, Lisa, and a big hand to Kevin and Afton. That this is, it's been a long time coming and a lot of work. So I don't have a lot of time, so I'm really going to go through this kind of fast. And I don't want this to be scientific, but more storytelling. And I'm going to go through 3 different families, 2 real and 1 hypothetical. And I'm going to tell their story in this chart, where on the y-axis, we have something called the estimated glomerular filtration rate. Hopefully, most of you are familiar with this as a scale, which your number, let's say if you have 100, 110, that means your kidneys are doing pretty well. And on the lower axis, on the y-axis here, we have kidney failure, and we have the age of the patient. Now, normally for, let's say, a healthy person, because your EGFR, age is part of that equation, your EGFR is always going to go down, even if you have perfectly healthy. So you'll start somewhere maybe around 100, and then by the time you get to, if you make it to the age of 100, maybe you'll be in the low 60s or something by that. In comparison, here's an X-linked male with a COL4A5 mutation called C.4688G grade A. Don't worry about this. This is a kind of a typical X-linked male. In fact, this is I'm this X-linked male in this story for this reference. And I had kidney failure when I reached the age of 22. So my EGFR started, I was healthy, and by the time I was 22, I went below that 15 of EGFR and started dialysis. I'm not going to go into the details of where my transplant is, but I do want to show here's an X-linked female who happens to be my mom. Same exact mutation as I do, but because she has 2 X's, the progression for her is slower. Here's another patient, which I have. This is a female with autosomal recessive Alport syndrome and has a mutation on the COL4A3, COL4A4. So even though we typically have X-linked males, will have typically, but not always, kidney failure at a younger age. In this case, it's a woman with kidney failure at a younger age. Here's an X-linked male with an X-linked mutation that's not as severe as the one that I have. It's a different variant, sort of a different flavor. Autosomal dominant female with a less severe, but she started with, for some reason, more impaired kidney health. I'm going to go, here's an autosomal dominant male who, looking at if they have kidney failure, may not be until they're in their 80s or 90s if they live that long. A digenic female who has, mutations on both the X and the 2 chromosome, severely impacted, looking at kidney failure in their early 20s. And I can go on and on and on. We think there are tens of thousands of potential of types of Alport. And so when we go look at this, when we're doing research on it, if you take all these tens of thousands, we have to make sense of this in a way. And one way we do this is we sort of put them in 2 very large buckets. We have fast progressors and we call slow progressors. And really take those quotes around it with a big grain of salt, because there are all sorts of exceptions to these types of rules. And this is the wordiest and scientific slide I get into. And I'm happy to take questions or share this slide with you. So when we talk about fast progressors, we're usually, but not always, talking about X-linked males, autosomal recessive, either boys or girls, digenic, and sometimes even trigenic, either boys or girls, with what we call more pathogenic variants. And I'm not going to go into the details, but this is how your DNA, how the mutation is actually impacted into it. It could be missing 1 letter, it could be missing a whole chunk of letters, it could be cases where the whole collagen gene is missing. And then on the other side, we have the slow progressors, who are usually, but not always, X-linked females and autosomal dominant, either men or women. And these could be with less pathogenic variants. So we have this whole different group of where we have the fast and the slow. One very important thing we say, in the past, we've seen that these slow progressors, the X-linked females and autosomal dominant history, were called carriers, or called thin basement membrane syndrome. That's a big red no. And then, we get on our platform, if we hear someone say, oh, you're just a carrier, or you have thin basement membrane disease, that is incorrect. We do our best to sort of stamp down on that and say, you are Alport syndrome patients. But you can then sort of classify, oh, I think I have a slower version, or we have the faster version. Okay, that's great, Andre. That's a lot of information I just threw. What should I make of this? What am I? Am I a fast progressor? Am I a slow progressor? And if so, how fast or how slow? Well, the answer is we really don't know. but we're trying to figure that out as quickly as possible. And this is where participation in the Alport Syndrome Alport Patient Registry, which was just relaunched this August, and Dr. Yee, who's going to be talking about the Neptune Ancillary Study. What are we doing with these? By analogy, let's say you have a genetic test and you have your latest kidney renal panel. You know what your glomerular, you know what your filtration rate, your BUN, your creatinine. Think of that data as being a GPS reading. Not only is that GPS reading going to say, oh, where am I on the map, but it also helps us construct the map itself. We don't have that full map. We're making it up as we go along to understand we need to know where everyone's own journeys have been as patients or as your kids have patients. So what we're doing is we're constructing the map and also placing ourselves on it at the same time. So I'm going to return to my family case and start making a few exceptions and then go into these other family stories. So one thing is my mom, she wasn't able to tolerate a high dose of ACE, but she was able to tolerate a high dose of an ARB blood pressure medication. And one thing we know for ACEs and ARBs, if you can tolerate the highest tolerable dose of that, it is known to buy you extra time. So my mom started on an ARB about 25 years ago. We're pretty certain that actually gave her, or has given her, I mean, she hasn't reached kidney failure yet, but one of the things that has improved her kidney function numbers too, that she got more kidney function than she would normally expect. I also have a nephew who has Alport syndrome, has the same variant as I would, but we knew when he was born, we knew he had the same variant as I did. So immediately, he had the luxury of saying, hey, can you put you on this blood pressure medication? There wasn't that knowledge when I had it. So now that he's on a high dose of ACE and he can tolerate it, our hope is that instead of his kidneys failing at the same age as mine, he won't see kidney failure until he's maybe in his 30s. We don't know for certain, but this is the kind of information that the registry, his data in the registry will start collecting in that so that we know that he's on an ACE and compare that to my story and how my kidneys fail. It fills in a section of the map. Here's another family, X-linked male, X-linked female, same mutation. Again, the same kind of pattern where the son, in this case, is progressing faster than the mom. One thing that the mom, very recently, there's a class of drugs called SGLT2I inhibitors, or flozins, that have come onto the market that are known at this point. Adults can take it. We're working very hard with the FDA to try to make it available for pediatric patients. But the mom has recently started on SGLT2I, and we're hoping, watching her numbers, to say we think that's going to give her some extra kidney function. Unfortunately, when the male was progressing there, looking towards kidney failure, it actually turned out, hey, there's a clinical study that's available you can enroll in. And we don't know what that's going to mean. So the patient, in this case, the son, said, okay, I'm going to participate in this, but I don't know if it's going to improve my kidney function or if it's not going to do anything at all. The one thing that was guaranteed to him now is that we knew if this study did something that took his kidney function and made it progressive and faster, that the study would be stopped. So when he entered the study, it wasn't known if it would work for him or not. So he participated in this. This is a way that I can help the research to figure out this map. I'm not doing it for myself to understand, this is going to cure my kidney or not. So he sort of took a risk to understand the landscape. In the end, he participated in the study. The study was not successful. And what I want to say is that it didn't deteriorate his kidney any more than it would have been if he had not been in the clinical trial study. So an important thing to understand about clinical trials in this case is you don't know if it's going to give you extra. You might be in a placebo group, but at the very worst, you're not going to progress any faster or slower than you would without being in that clinical trial. So here's one more family case for a story. This is a hypothetical, and let's say it's going to be a long-term story, maybe over 50 or 60 years. We have an autosomal dominant Alport syndrome parent. In this case, it's a father who has a mutation on just the COL4A3. And we have an autosomal recessive Alport syndrome child. Let's say it's a daughter who we know is a fast progressor, looking at potentially with the mutation they have, we believe it's a COL4A3, a COL4A4, that they're going to experience kidney failure by the time they reach 20. But because we caught this early, and we know the parent can't take an ACE or an ARB and they're on SGLT2, but the daughter in this case was put onto an ACE inhibitor early and was able to tolerate it. So we hope, we believe that the patient won't go into kidney failure probably until their 30s. Around the time the daughter turns 20, a clinical trial came up. They enrolled, same with our last family in this case, but it wasn't effective. So they're still on track for, we think, maybe kidney failure at best at the age of 30 with that ACE. But another clinical trial comes up when they reach the age of 30, and they're still good enough and they're effective on that. In this case, the clinical trial was effective. And we're talking over a period of like when it started from maybe 10 years or 20 years. So we're talking at the age of 30, watching what the numbers are. We're predicting that on this testing drug, we say, hey, we've basically halted the progression of use, so we believe that you can go on this, receive it as a drug, and the FDA approves it. You're now not looking until kidney failure until you reach maybe the age of 60 or 70. With the FDA looking at the study and say, yes, we're approving this, now it's available for all Alport syndrome patients, including the parent. So now the FDA has approved it, and the parent gets a prescription for this from their doctor saying that now what the parent is looking at, instead of expecting kidney failure at the age of like 65, now it's like you probably have good native kidney function for the rest of your life. Finally, at some point, even further in the future, there's another clinical trial. We'll call it Clinical Trial X. Neither the father nor the daughter are in that trial, but the trial turns out to be successful for the participants of that. The FDA approves that drug and says, hey, the daughter in this case, you still haven't had kidney failure. We're going to put you on this new drug. And that actually buys them even more time. So they're not worrying about kidney failure until they're maybe in this case 90. So this is kind of, again, a hypothetical situation where we're talking about 3 different clinical trials, only 2 of which the daughter in this case participated in, and a 3rd clinical trial which led to a successive drug, which in this case the daughter was able to take. So I wanted to give this as a high-level overview of the importance of clinical trials and why we want to progress. That if you're available to enter a clinical trial, it helps the larger landscape understand, will this work for not just for me, but work for you? And we keep this updated on where there are clinical trials on the Alport site to update on. So I want to say, let's consider maybe 30 years or 20 years from now, there is a special class of clinical trials that are not there now, but we're hopefully which are called genetic therapies, which we'd hope is if they can deliver them to that, they're actually curative in potential. And with that, and we're sort of going to say that in this case, we're 20 or 30 years in the future, and we've got this drug, and in this case, both the parent and the father and the daughter in this case receive it, and it basically cures their Alport syndrome. Again, this is hypothetical, and it's basically going to put their kidney function so that it doesn't progress fine, but it basically puts on a hold so they're essentially healthy. And the real goal here is, let's say there's a granddaughter that comes up at that point. We know that granddaughter is going to be born with Alport syndrome. We start treating that grandchild as soon as possible, and eventually the hope is that that child will basically be cured of Alport syndrome by receiving that drug. So that's the long-term hopeful landscape what we're doing with, and it sort of gives the idea of the registry, again, is the idea of where do I fit in that landscape, and then the importance of where clinical trials are going on with that. Again, thank you. Picture of my nieces and nephew, and again, thanks for taking questions later.
All right, and the last piece, before we take a little break, before our next panelists, which get into all sorts of managing all aspects of Alport syndrome. Our last speaker is Dr. Jennifer Yee, who came all the way from the University of Michigan. She's a pediatric nephrologist and research scientist at the University of Michigan. Her specialty interests include proteinuria kidney disease, especially genetic forms of glomerular diseases like Alport syndrome. She completed pediatric nephrology training in both Taiwan and the United States, and earned a master's degree in public health at Harvard University and a PhD degree in cell molecular biology at the University of Michigan. She's a co-investigator for the natural history study in Alport syndrome as part of Neptune at the University of Michigan, and that natural history study is funded completely by Alport Syndrome Foundation. We're honored to do so, and André spoke a little bit about it, and Dr. Yee is going to give you all the details. Thanks.
Hi everyone, I'm Jennifer. When my friend asked me, what do? I usually tell them I take care of kids with kidney problems and also teach them how to understand and how to protect their kidneys. So I'm here today to talk about natural history study in Alport syndrome. So it is a study is supported by Alport Syndrome Foundation. It's affiliated with other patient-funded kidney organizations, and now it's incorporated into the Neptune study, which I will introduce later. So what is Neptune study? It is an abbreviation of Nephrotic Syndrome Study Network. So it is a patient longitudinal cohort study to study patients has massive proteinuria, leads to whole body swelling and all kinds of complications. And it was established more than a decade ago and includes 32 North American sites recruiting patients. And now, the natural history study in Alport started to enroll patients through this Neptune study, applying the same study strategy and structure, collecting biosamples and clinical data, including some audiology examinations with intention to find a non-invasive biomarker and therapeutic targets. So this is the map for the Neptune study, which has 32 North American sites, and more than half of the sites right now started to recruit Alport patients. For example, in the Washington State, there's a Providence Research Medical Center, Seattle Children's to recruit pediatric patients. A couple of them in California, East Coast, in the middle, there's a Kansas City, Missouri, Ohio, and Michigan. And in the south, there's Texas, Carolina, and Georgia. And why this study is important for Alport community? Because it uses a unique approach in which, in addition to collecting clinical information, it also collects very extensive molecular information from the kidney biopsy change, your gene, your mutation of the collagen 4A gene expression level, protein and metabolites from your blood and urine to generate a multi-scalar data. Eventually, we want to integrate this into the clinical outcomes. So we anticipate this approach can help us to discover non-invasive surrogate markers and also therapeutic targets from investigating the disease mechanisms a little bit better. The long-term goal of this study is to understand what causes the kidney progression in the Alport syndrome. We would like to discover non-invasive surrogate markers, so everybody doesn't need to go to a kidney biopsy. And we want to identify the shared and specific disease mechanism of different collagen 4A mutations. So we know that some certain mutation tend to have more faster progression of a kidney disease, and some result in a milder form. And we're kind of hoping to know the mechanism a bit better so we can shift the severe form back to the milder spectrum of the disease. Eventually, we want to use this approach to develop clinical trials that target the candidate mechanisms. And hopefully, we'll use the right trial for the right patient at the right time. So who is eligible for this study? So basically, everyone with Alport syndrome is eligible for this study. And the research visit is every 4 months up to a year since the baseline visit, and after that it's every 6 months up to 3 years. And as I mentioned before, other than some clinical data, your geographic factors or information, we also collect a lot of biosamples from your blood and urine, and also sequence the collagen 4A mutations and all kinds of other different gene expression level. So this is a flyer in the Alport Syndrome Foundation website. So the study period is 3 years. You can get more information from that. And then if you want for more information, or you are interested in participating in the study, there's an email over there. You can just email this address, and then somebody will, get contact with you again and kind of allocate which nearby institution you can go to. So it's a short talk for me, but I think every one of you here can contribute to the medical research and help researchers better understand diagnose and treat, and eventually with a hope to cure Alport syndrome. Thank you.